Hello everyone, welcome back to our video on gene interaction. In the last session, we discussed about the variations of, we were discussing about allelic interaction. And in allelic interactions, we were discussing about we discussed about the first type of allelic interaction, which is the allelic interaction. And in that, we discussed the variations in dominance. And in variations in dominance, we came across the complete dominance and some of the deviations from normal complete dominance, which where we discussed the haplosufficiency, insufficiency and dominant mutation. And now we are at the second type of variations of dominance, and this one is called as incomplete dominance. So, from the name itself, incomplete dominance, it states that the dominance is not complete. The dominance is not complete in the sense that here the alleles which are interacting, they are neither dominant nor recessive. You cannot term them as dominant or recessive. So here what happens is the phenotype of the heterozygote is intermediate between the two homozygous or homozygotes. So we have two homozygotes as parents. So one is homozygous red, as in the case which is shown here. As you can see here in the diagram which is shown, as you see here, you can see here the homozygous, one of the homozygous parent which is red flower, and the other one which is a homozygous parent which is white flower. So there are two homozygous parent, and when a heterozygote is formed in the F1 generation, you can see that the phenotype of the F1 generation is a mixture of the homozygous parent. That means you have a mixture of red and white, which is a pink color. So it is an intermediate between red and white. And that is what is called as incomplete dominance, where neither of the homozygous parent can be termed as dominant or recessive because a mixed phenotype appears in the F1 generation or in the heterozygote. And heterozygote always displays a mixed or an intermediate phenotype of both the homozygous parents. So in incomplete dominance, each of the wild type allele produces. So how this intermediate? How can you explain this incomplete dominance? And this is explained by the dose by which the particular pigment is being produced. In this case, it is regarding pigment because it is the flower color which we are discussing now. So, in this, we are going to discuss the pigment production, how the pigments are produced, and at what rate the pigments are produced in this particular case. So in incomplete dominant, each wild type allele. So here you can term something as wild and the other one as mutant rather than dominant and recessive. So in this case, red will be the wild variety or the wild type, and white will be the mutant phenotype. In the sense that always wild will be the common color or the common one in the population, and this one the mutant one will be the one which will be seen in less numbers in a population, and that's how you define a wild type and a mutant variety. So a wild type, in this case, in this particular example of incomplete dominance, is the red color flowers. So this particular wild type allele, the allele which is present in the wild type, produces a dose of a particular protein. So protein that may be an enzyme, and that enzyme may be responsible for producing a particular pigment. So when that particular enzyme is present, a biosynthetic pathway may be catalyzed by that particular enzyme, and that will result in the production of that particular enzyme which give particular protein or the pigment that will give you the color of the flower. So the number of doses of the wild type. So here what happens is the number of doses of the wild type determines the concentration of the chemical which is or the pigment which is made by the protein. That means when you're having a homozygous uh, wild type plant, homozygous wild type means it will have two doses of the wild type allele. That means here you have two capital R's here. So in this case, when you have two wild type allele, the amount of that particular enzyme produced will be double because each allele will be producing or will be transcribing that particular enzyme. As a result, you get more number of enzyme. When there is more number of enzyme, more pigment will be produced, and that result in the color which is called as red. So, when you have a double dose of a protein or a double dose of an allele, not the protein, double dose of the wild type allele, you get double dose of the enzyme and hence double dose of the pigment. As a result, you get the color which is called as red. When you come to the mutant, which is a white variety, in the white variety, the enzyme might be not present in sufficient amount, or the enzyme might be a mutant. So in that particular case, in the white plant, what happens is that this particular biosynthetic pathway for producing the red color is not active, and hence the pigment is not being produced, and hence the color is white. So this is how the color or the mechanism by which the color is produced. So in this particular case of white flower, there is no color formation, or there is no something which is called as uh, pigment formation. So here there is one mistake which is made here. So just take care, because this image I have taken it from the internet. So in this image, you can see that one is given as R and the other one is given as W, and they have shown this to get an idea. They have avoided the usage of small r, because there is to show you that there is no relation ship like capital R and small r, where dominant and recessive is present. But you should remember that usually when you use two alphabets to represent the gene, that indicates that they are two different genes. Okay, this might, or you might take in mind that as these are two different genes. Actually, these are not two different genes, because we are right away talking about what is called as allelic interactions. So in allelic interactions, you can mark it in another form. That is, you can take it as capital R. Both can be represented as capital R itself, as there is no what you call a dominant and a recessive relationship. So both red and white allele can be taken as capital R. You can put a plus sign. So this is one form of representation. You can put a plus sign for the wild type, and you can put a minus sign for the mutant variety. So what happens is, you will have such a combination of red and white. I'm making it or I'm putting it in a slow manner, because I'm doing it right away in the laptop, but I'm not in a speed to control my mouse like this. So this can be taken as a small capital R negative. So in this particular representation, you can see that there is no such relationship where one is dominant and the other one is recessive. Plus refers to one which is wild type, and the other one refers to the one which is the mutant variety or the white variety. And in the F1 generation, what you can represent it in this particular manner, which I'm writing it now. The one will be like this. The heterozygote will be like this. Because they are alleles, you cannot take them in different alphabets, so you can take it like this. So this will be the heterozygote, which will give you the pink color. So these and from this you can derive the other ones also in the F2 generation. Now, so you can use this particular alphabets rather than the one which is given in the diagram, so that you don't make or you don't confuse with the coming sessions, which is the non-allelic interactions. So this is an allelic interaction. These two are alleles, but they lack a dominant and recessive relationship. So this representation might be confusing when you look back after a month or two. So you can take this particular alphabets to represent this particular example of incomplete dominance. So what we are talking about is about the dose of the protein. So when you have 2 plus R plus, you have a double dose of the pigment, and when you have this minus, you don't have that, or that pigment is not being produced. And what happens in the, and two doses in a homozygous produces a greater amount of the pigment, and there is no dose in the case of a homozygous mutant variety. As a result, there is no pigment. And when a heterozygote is formed in this particular case, yeah, what you can observe is that you have only one R plus, which give you the pigment, which is half of the homozygous one, and the other one is R minus, where you do not have the pigment production. As a result, you will get a color which is intermediate, because only half of the pigment is being produced, the other half is not being produced. As a result, an intermediate color was observed in this particular case, and that color is called as the pink color. So, which will be one dose less, or one dose less in the heterozygote produces less pigment, so that the petals of the flower of the heterozygote appear pink. And here there will be a change in the phenotype or phenotypic ratio. The normal Mendelian monohybrid ratio was 3 is to 1, the phenotypic ratio, which was 3 red. In the same pattern, Mendel defined it as 3 is to 1, which is 3 red is to 1 white. But now it has changed. In incomplete dominance, it will be changed to 1 is to 2 is to 1, which is 1 red is to 2 pink is to 1 white. So this is the changed or the modified Mendelian monohybrid ratio when you come to what is called as an incomplete dominance. So both the phenotypic as well as the genotypic ratio will be the same, which is 1 is to 2 is to 1 for incomplete dominance. So an example of this is the 4 o'clock plan, what you show here. So what you have to remember is just take this one as the take this as your representations for when you write incomplete dominance. So that's all with incomplete dominance. So in incomplete dominance, what happens is there is no complete dominance as said by Mendelian monohybrid or Mendelian dominance. So here the dose of the allele or the number of allele depends the phenotype. So here the number of alleles when at us in a heterozygote you have only one dose, only half of that particular pigment is being produced, and that will result in the production of lesser number of pigment. So I might represent this once more. So what you have to do is you have to take it as R plus and R plus for the red parent and R minus and R minus for the white, and heterozygote can be represented as R plus and R minus. Okay, so this one will be your red parent, this one is a white parent, and this one is the heterozygote. So you can, when you write the example, you can code these alphabets so that there is no confusion regarding whether it is an allelic interaction or non-allelic interaction. This one is definitely an allelic interaction. So coming to the next one, the other one which is called as the codominance, which is a third one. So in codominance, so here also from the name itself, we can derive what type of interaction it might be. So here in the previous example of incomplete dominance, we saw that only the wild type allele is being expressed, and the expression of the wild type allele depends upon the dose, and that dose depends and that dose determines what phenotype it produces, which is an intermediate between the two homozygous parent, homozygous wild, and homozygous mutant. But here codominant means here both the parent phenotypes or both the homologous or both the alleles of the parents, they become dominant at the same time in the heterozygote. Or when a heterozygote is being formed, what happens is there is a co-dominant character. That means the expression of both the alleles can be seen in a heterozygote, and that is what is called as a co-dominance. You can see both the phenotypes in the same individual in the heterozygote. So they are equally expressed in the case of, you can see an equal expression of both the alleles. And in heterozygote of codominant nature, both the parental traits appear side by side. That's you can see both the phenotypes at the same time in the heterozygote individual. So both the alleles become codominant at the same time, and each allele has the capacity to be equally expressed in the heterozygote. So here you can take and the example as here is you have the same example. We are moving on with the same example here of the plant. You have a wild type variety which is red color and the mutant variety white. So if these alleles in this particular plant, it expresses or it shows the condition called as codominant, you can see a condition like this. That means you can see both red as well as white being expressed in the same color. If it was in complete dominance, a complete color or the complete flower would have this would have a single color which is pink, but you can see this is a co-expression of white and red, and this is what is called as co-dominance. And this is more prominently seen in cattle, horses, etc. So in cattle, you can see when you have a wild variety of parent which is red in color. Red means dark brown in color, which appears to be red, and the other one which is wild, or the white one, which can be considered as a mutant phenotype. So when wild and the mutant one is crossed, mutant in the pigment formation is being crossed, you get a round coat color. So you can see both the parental phenotypes or parental phenotypes are expressed in this particular heterozygote. And this particular condition is referred to as codominance, where both the alleles of the parents get equal expression or equal phenotypic expression in the heterozygote, and that is what is called as codominance. So, codominance you can observe in such a patched manner. This is patches of both the parents. You can also get in the round color as a single coat rather than patches. You can see the lighter shade than this. So in such animals, if you observe their hair, if you closely observe their hair, they will have both the hairs in them. That means it is said that half of the hairs will be the red in color and the half will be white in color. So there will be an intermediate or a mixture of hairs, and that will give you a lighter shade than this. So you can see round coat color like in a patched manner and also in a single color manner. The single color will be represented, or a single color, how a single color is formed is that in that particular animals or that particular cattle, horses also, you can see this particular phenomenon. In them, you can see both the type of hairs will be present in their body. It will be in a mixed pattern, not as patches as you see in this particular case. It will be mixed. Both the red color as well as the white color hairs will be seen in a they will grow in a mixed pattern. As a result, they will get a lighter shade than red. If you closely observe their hair, you can see both red color hairs in them as well as white color hairs in them, and that is what is called as the roan color. So both the phenotypes is having an equal dominance, or they appear equally in the heterozygote, and that is what is called as your codominance. So in codominance also you can take this as an example of codominance, as here it is represented as capital R raised to small r, which is red, and raised to W, which is for white. And you can see this, and when in the F2 generation you get a phenotypic ratio which is 1 red is to 2 uh, round color is to 1 white. So this is also a deviation. So this is similar, the phenotypic as well as the genotypic ratio of the F2 is the same in this case, as well as the incomplete dominance, which is 1 is to 2 is to 1, which is a deviation from Mendelian phenotypic ratio, which is 3 is to 1. So 3 is to 1 normal Mendelian ratio is now changed to 1 is to 2 is to 1 in the case of incomplete dominance as well as for codominance. So this was the other two variations of dominance, which is incomplete dominance and codominance. We have one more, which is lethal genes or lethal alleles. We'll discuss this lethal alleles and lethal genes in the next session. Thank you for here.